Welcome. I'm Liz Taylor, ocean explorer, naturalist, and entrepreneur.
I'm Sylvia Earle, explorer, National Geographic, founder of Mission Blue, and I'm an ocean elder. Thank you, ocean elders, for sponsoring this program.
And this is our show Dive In, where we host informal and open conversations with the ocean community on topics of wonder and interest. We're very glad that you've taken time to be with us and hope everyone's had some time to get outdoors.
Yeah.
You can ask questions by putting them in the Q&A box, and we'll get through to as many as we possibly can towards the end of the program. And before we get started, let's remind everyone that the world
Is blue.
blue. Today, we are so excited to have Dr. Barbara Block with us. She is a world-renowned scientist who focuses her research via tagging on large predatory pelagic fish, such as tuna and billfish and sharks. She holds the Charles and Elizabeth Prothro Professorship at Stanford University and earned her PhD at Duke University, just like you.
Yes. We're Blue Devils.
Blue Devils, yes. You really are, a pair of you. Um, and joined the Stanford faculty in 1994. She's received a MacArthur and Pew Fellowships, the Rolex Award for Enterprise, and the Benchley Award for Ocean Science. Welcome, Barbara. We're so happy to have you with us.
Well, welcome to being together with Sylvia and Liz and having this opportunity to talk to both of you about the blue ocean.
Yes, yes, absolutely. Sylvia famously says that her introduction to the ocean came when a wave just like toppled her over when she was about 2. Um, how did you come to become entranced with the ocean?
Well, I think like many ocean lovers, we have to credit our parents. And so I grew up in Massachusetts on the shores of Cape Cod and just was introduced at an early age to beaches. And I can remember digging holes with my brothers, trying to find, the other side of the earth and burying rays and parts of sharks and, doing all the things kids do.
Yeah, yeah, all the beachy things when they have the opportunity. That's wonderful.
Yeah, right
And
there.
I think swimming is a good, if you're given strong instruction on how to be safe in the water at an early age, I think it builds confidence that makes you love the water. So
Yeah,
I credit my parents for pushing us to be good
water
swimmers.
water competency is just such a core thing. I really, I mean, we did it with our kids, and I certainly got thrown in early. That's excellent. Um, so you have some slides that you're going to share today, and maybe we'll talk about, a little bit about your early career at Woods Hole and your time with Dr. Francis Carey and leading to the Census of Marine Life and beyond to animal tagging.
Great. Do you want me to just get started? I did
Jump
start.
in. We're just
No,
gonna,
no,
we're just
I
gonna.
did start way back at Huey, so I have really good ties to Massachusetts. I can look straight out in Monterey Bay right now and see the ocean blue. So what I'm going to do today is tell you a little bit about how we study animals in the wild. My team here at Stanford, we study 3 groups of fish, the
Mhm.
tunas, my favorite, of course, the
Absolutely.
white shark and the lamnid shark. So that would be the mako and the porbeagle and salmon sharks. And then I'm also heavily tied to the billfish. And what all of these groups have in common is, I'll show you at some point, is they have the ability to keep their tissues warm. And my training is as a physiologist at Duke with Knut Schmidt-Nielsen, one of our favorite physiologists on the planet. And my interest really is in how these animals use that warmth to actually explore the planet.
That's such a cool thing. We always think of fish as just being, most people think of fish as being sort of
Cold-blooded.
cold as a fish.
Yeah, and to tell you the truth, no pun intended, thermogenesis is hot right now. I'm not going to tell you about how muscle makes heat, but believe it or not, it's really an active field. And I'm giving a talk at a medical school in a week or 2 about how muscle makes heat. And I'll tell you why that's important in a minute.
Yeah.
Here's where I am. I'm going to just point for the listeners. I'm actually at Hopkins Marine Station. I'm right in the corner building right there. I'm looking straight out here. We live in
Nice.
one of the prettiest places on the planet. And one thing I like about being at Stanford at the Marine Station is I'm not at main campus. I see wild animals right outside my window all the time. And I do have this fondness for being at sea. Now,
Yes.
as Sylvia knows in particular, I got started out here because the aquarium wanted to keep tunas. I hope many of you have come to see the beautiful
Oh,
tunas
yeah.
at the Monterey Bay Aquarium.
Oh, incredible.
And we built the only tuna center on the North American shores right here at Monterey. And we're now discussing renovating it. So it's 30 years old. And
Wow.
in this center, we were one of the only teams to bring tuna into captivity. First, display them at the aquarium, which was a great accomplishment by the aquarium and Stanford together. And then our real story was we brought bluefin into captivity, and that's one of our bluefin tuna there. And when you're behind glass and you can see this animal, you can see how gorgeous it is. Has a streamlined bullet shape. It's got a naked skin, so it doesn't really have scales for 2/3 of its body, and that makes the fluid move across the body without having too much drag. And
Hmm.
then you can see some scales here, like in a golf ball. There's some bumps there.
Oh,
And
yeah. Look at that.
it channels the water. It's just amazing. So I love having the bluefin on display. You see the beautiful high aspect ratio tail.
Mhm.
And for me, as a tuna lover, it became really apparent we could do a lot having these tunas on display. Now, I had to show you our biggest one. I haven't shown this picture in a while, but we did grow them as large as 580 pounds. Sorry about that, Sylvia, but one of the problems of keeping bluefin is they overgrow the facility. And so that was really a bit of an issue early on. But like Sylvia, what I really love are the wild bluefin. This is an Atlantic bluefin. And I'll start by saying Sylvia can be happy. We brought them back, and they're back in all three populations. So there's three species of bluefin, the Atlantic, the Pacific, and the southern bluefin, and all are doing pretty well, despite the fact that people do like to eat them as sushi. That's how
Yeah.
most people think of tuna. But as I mentioned, I think of tuna very differently. They're one of the rare endothermic fish. So what they do is they move their swimming muscle that powers them across ocean basins. It's a dark red meat, much like the dark meat of a chicken. And in
Wow.
biology, where you see that red, it means there's a lot of heat production. We, in a class once, took an animal that we had sacrificed and took a picture with a infrared camera, and you can see the heat from the animal. So the other specialization is the animal swims with a unique form. It's stiff swimming, and it actually has an elastic set of tendons that enable it to literally swim efficiently across all ocean basins. Now, for your viewers, most of us think of warm fish as mammals and birds. So here's a penguin,
Yeah.
and you natively think of it as being warm. Here's some seals, here's a whale. And we call all of those animals who make heat from within endothermic animals. But less known is of the 40,000 or 35,000 species of marine fishes, there's actually some families that have endothermy or heat from within as a trait. And that includes the tunas. And so the heat is actually warming their eyes and their head, their viscera, and their muscles so they can swim efficiently across ocean basins. Here's a porbeagle shark. No, that's a salmon shark. So the
Yeah.
lamnids, the white sharks are warm. And then little known is the fact that many of the billfishes have a brain heater. So the swordfish has a very big
Wow.
brain heater, and that makes heat beneath the brain. And then most recently, students associated with my lab and others have found that OPA is an endothermic fish. So these are sort of our four groups. There's actually a few more out there, but about 50 species of endothermy. And because those animals breathe with a gill, they tend to like have the window open for heat. They let the heat out
Yeah.
into the ocean.
It's so cool. And the swordfish has like such an enormous eye too, and it's interesting that it's got sort of heating the brain proximate to the
It's
eye.
actually heating the eye. It's actually got a big tube of blood going right to the retina. So imagine they're in a physiological war with squid who can escape with their giant axons really
Yeah.
quickly. And then the swordfish is down in the deeps and the depths. It's very dark. And by having a warm eye, it's hypothesized they can actually pick up a flash of light in the mesopelagic depths at high precision for a vertebrate,
You,
a backboned animal.
you've seen them from the submersibles too, haven't you?
I have, yes.
Yeah.
So here at the lab, really written the book on tuna physiology, and we almost had tuna spawning here a few years back, right in the midst of COVID. We've been able to grow them up and trying to figure out what makes an amorous tank where the tunas like each other. It's pretty hard because you don't know who's male and female, although recently we came up with molecular technology that allows us to do that. Those are tunas not in the aquarium, but in our Stanford tanks. And in the Stanford tanks, before I get to the wild work we do, I want to say that having tunas in captivity allowed us to do some really fascinating things. So one of our postdocs, Vadim Pavlov from Ukraine, he figured out one day by watching tunas that they have a hydraulic system. They're the only vertebrate who can actually raise and put down their fins with fluids.
Hmm.
We had other students who came up with ways to put cameras on the tunas, and then we took them out to the wild. And it was here we discovered that the tunas actually often pause in their swimming and are drifting down with gravity and using elastic energy more than we realized. And we took the ideas of how tunas move in the wild, and with some scientists from back east, we were able to work with them on their mechanics of making a surveillance tuna.
Robotuna.
And I threw this one in for Sylvia because we just got this picture. Wait till she sees it. This is a tag that's taking all sorts of data. The data strips are how the animal's pitching, how it's accelerating, but this is the greatest shot. You feel like you've seen Amelia Earhart's airplane down here, but that's the fin of a 1,000-pound bluefin carrying the camera, shooting its
Wow.
buddy, a picture of his buddy over here. So that gives you some idea of what we do. What I really like to think about is that we go from molecules to whole animals, from genes to ecology. And what we're trying to do is understand what's the future of tuna in a warming world.
Yeah.
How is it their biology and their physiology will provide resilience as we change the ocean. So some of you may wonder, why do we want to even study these animals? I have to remember how to get rid of my top here, but I can't. Let's see.
All right. It's yeah, the heat anomalies that are just starting are just, they're really frightening.
Right.
How hot
Yeah.
yeah.
And I think most people don't think about how cool most of the ocean typically is. That we're pretty much surface oriented. But
That's right. So this is a map that's showing us the heat anomalies where it's hot. So one of the things we've been worried about in the future is what's the role of physiology in helping these animals when it gets hot?
Hmm.
And how is it that they're going to avoid that? I'm trying to get that screen thing out of the way, but I can't. So for some reason, I am stuck, but don't know why. There we go. I wanted to show you the coolest new piece of biology data. We put heart rates in wild tunas. We only have two back. Down here is just like you looking at your watch, a heart rate of a tuna in the wild. There's a week of data here,
Hmm.
night and day and night and day. And this is 50 beats per minute. And you can see the animals going up to about 70 and then down to 50. And when it dives, I'm not going to get into the detail, the blue are the dives. We're seeing heartbeats as low as 20 as the animal goes into cold water that's about 10 degrees C.
Wow.
So unlike the mammal, like Barb Block or Sylvia Earle, who keep their heart rate high all the time, the tuna's got flexibility in that it can go in the cold and let its heart rate drop. But then up in the warm water, it's got a heart rate just like us, very high for a fish.
That's
And
interesting.
then I'm going to tell you one last, just how do we get science from the lab to the field? In the field, we can measure where a tuna goes. We'll be talking a lot about that. And in the lab, we measure how much energy the tuna eats. So today we're plotting how many kilojoules are ingested by a tuna. We got that from the lab, and then we took it to the field because we're measuring the tummy heat in the wild. And so this is a tuna who is getting many more calories. So this is calories per day right here, ingested per kilogram. And what you can see is in the spring, it's to the left, and in the summer, it's to the right. Colors are hot when they're getting lots of food. So this might explain why tunas migrate. They might come to the California, get a lot of food, and then go down towards what we call the intertropical convergence area, where they might get less food, but still make it on diving into the mesopelagic. So what I've shown you just a little bit is what we do in the lab, but what really matters is how do we observe these ecosystems in the wild? And what we like to do is think about the challenges we all have. I'm looking out my window, and Sylvia and Liz could be looking out theirs. And all we see is an empty sea. There's no transparency. So my career has been about working with students to help us figure out how we can study where these animals go. And I think what I want to say today is that it's really hard as primates to understand the fluids and the physics of the ocean. And so I feel like where we've made a huge breakthrough with the biologging technology I'll be telling you about is that we really don't, as humans, as primates, understand what it means to have chlorophyll at the surface. That's what's shown there, or what it means over here to see fluids moving. That would be physics. And these animals are better oceanographers than we are.
Isn't that often the case that, the nature's really kind of figured it out and we're kind of coming in a little bit later and going, oh, we've just discovered this, but nature's been doing it all along.
Yeah, I like to say the white sharks are way better oceanographers than we are. So what I and many people who do the field that I'm in called biologging do is we try to figure out how the predators move here off the California coast or the Atlantic coast, in this case, tunas and sharks. The tools that we're using at the local institutions are the same tools across the planet. We use tags that provide information on the ocean. We use gliders and drones. These are automated vehicles that help get data. Very hot right now is genomics. Our lab is part of the genomics revolution in the ocean. We like to do a lot of it. And then you can pick up a water, and just like a forensic scientist, you can figure out who swam by, and we're doing a lot of white shark environmental DNA. That's a pretty
Yeah.
hot thing.
Yeah, those guys like to just kind of slink by in the murk, It's hard to spot them.
So what's exciting now is that the field of biologging has been advancing. It's still a pretty expensive device when you put it on an animal, but we can actually take satellite tags. We can take tags that just take data. They're called archival tags, and put them on animals and figure out where they go. And we do it with a variety of technology, but it's all really based in mathematics.
Hmm.
So for a white shark or tuna, we might have a tag that talks to an underwater receiver. We call that an acoustic tag. We might have a tag that's on the dorsal fin of a shark that's talking to a satellite called the Argos satellite. And then the tag that I've been most instrumental in helping to develop and get out into the sea is called the pop-up satellite archival tag. It combines the ability of taking data with sending data to satellite, and then the camera tags I illustrated might look something like that. And the biggest problem is these are expensive devices, but the beauty that I like to show, Sylvia hasn't seen sort of the updated map. This is the points from our projects. We run 3 major projects tagging tunas. We call it Tag a Giant, so tagagiant.org. We still tag predators in the Pacific. It's called TOPP. And then our newest project is with the IGFA. We're doing marlin tracking. And what the challenge is, no one has really come up with a cheaper tag. That's been a problem. I've got one at the end here I'll be telling you about, but the price on these things is high because there's small companies making them. And that's what limits progress a little bit. I'll say that honestly.
It's really tough to scale. And then it's just the harshness of the environment. It's like, how do you make it really stick, be durable, deal with changing in pressure and temperature differentials and
We
all
should
of
be
this stuff.
prepared to subsidize getting to know the live fish instead of subsidizing killing fish.
That would help. Kind of shift the funding around a little bit
$2
would be great.
billion used.
Yeah, I know they really, it's been sad how little has gone into this, but it's been a progression, and we had to make them work. We did have years of mixed success, and now we're doing quite well. And then for everyone listening, it's really an interesting problem how you follow an animal in the sea. What's happening on our tags is something you probably would never imagine. We're measuring light and photons, and we can measure a sunrise event and a sunset event every day. And then we're using astronomical math to calculate local noon from the threshold of sunrise and the threshold of sunset. We know halfway in between. And then we have a really good clock on the tags, so we know exactly what time it is, where we are from Greenwich. And then using an old sailor's trick, we're using an ability to then calculate a position on a map. And that position is a probability of where the animal is. And from that, we make these tracks that then become the dots that you see on the map. This happens to be a giant tuna tagged in our favorite place in Nova Scotia. And it goes down into the Gulf of Mexico to one of your favorite places. It spawns right here. We know that from some of the hot temperatures on the tag and some behavioral data, and then it'll come back up and go right back to where we were. It will release the tag, and then we are able to recover the tag and get the second by second data, which is a pretty good trick.
So is it
Yeah.
like a galvanic release or an acoustic release, or how does it release eventually?
Good question. That's a metal pin, and the pin gets a current, and that current then corrosively releases the tag. It breaks the metal pin.
Okay. And then it like it's buoyant, so it comes to the surface and then you can pick it up.
Yes. And so we do a lot of picking up tags. We have a bumper sticker. It says no tag left behind. So we're really good at getting the tag to get the archival data. And we have a whole fleet of students and fishermen who work together to help recover the tags.
So where's the weirdest place you has a tag ended up? Is it, do you have one like in somebody's garage or?
Yeah, we have them in garages. We just had one in a white shark tag that was in the garbage pail when they
Oh,
found
no.
it. We're not sure why. And then we just chased one in Greece that turned out to have a great story on it. So we will go to no end to get a tag back.
Yeah, because it's all the data, right? You really,
That's right.
yeah,
So then these are typical tracks being rebuilt, and then we can put it on the oceanography the animals swam through, and for the first time get really, really good data on how animals use the ocean. I want to say that it's important in part because the animals themselves are contributing data to the international data sets. That's hard to imagine, but the tags have temperature devices, some have salinity devices, and we really lack data from the ocean. And it's data from the ocean that tells us when a hurricane comes by, its strength, and it's data from the ocean that tells us our daily weather. So I
yeah,
like to say Earth's physiology is what we need to know more about, in part because these are the four challenges on the planet. We're warming, we're losing oxygen as we warm in the ocean. Because of absorbing CO2, we're more acidic. And of course, we need to do more to make humans sustainable. And these are the issues, the challenges I present to our students at Stanford that we have to spend our time thinking about how to solve.
yeah, yeah,
And I want to just say before I turn it back to you is that one of the challenges that a lot of people don't get is we don't understand really how the ocean is changing as much as people think we should. And we don't have monitoring systems that allow us to really see what's happening, let's say, with the prey or the predators. Of course, coral reefs we know a little bit more about, but we're really lacking investment in ocean ecosystems, in the science of monitoring them so that we can better predict what's going on.
yeah, yeah. Because I mean, a lot of these animals are very long lived. They travel, huge distances. So the information that can be ascertained from the tagging programs, it just has almost endless applications, I
That's
think.
right.
Yeah.
That's right. And then the biggest problem is, I have to tell you the truth, we can't count fish very well, so we don't really know how much humans are removing, except we know they're removing a lot. And
Mhm.
as we progress towards a planet with 9 billion, we need solutions from taking animals from the sea. And
Yeah.
I bring this, as always, with me to any conversation about sustainability. One of the things that makes me wonder is how could humans have revered the bluefin of all species so much? The whole civilization in the Med was built around it. Countries that ruled the Med ruled the tuna. And the tuna was the salted fish that people ate for their protein in many cases. And so that's honored in these coins and this art. And yet nowadays, they really just trade it for money.
Yeah. It's just money. They're just turning wildlife into money.
Right. So what I want to explore is, can we live sustainably? Can we get away from eating wild animals? And then can we somehow come up with tools that change the game for protecting the ocean? And
Mhm.
our former student, Doug McCauley, who's now, of course, a well-known professor at UCSB, he made this gorgeous graphic with an artist. And what it's trying to lay out is the transition on land to an urbanized world happened many, many years ago, over thousands of years. And yet, here in the ocean, while there isn't a pristine place as a whole out there, we know a lot of change is coming right now. And so we have control this generation over what we do. We can't go back and get woolly mammoths this week. But the bottom line here is we can make decisions off our shores that impact our futures. So
Barbara,
oh, go ahead. Sorry, Sylvia.
you did, in a fairly short period of time, transition our impact on the land from fairly light touch to a really heavy touch in the last 200 years. But we've really only begun trans transforming the ocean starting seriously in the 1950s. And part of it is fossil fuels, the ability to power vessels, the ability to go into the sea to extract those, well, oil and gas and apply them, and to use these powered vessels with refrigeration, with new materials like the materials that are now used for nets instead of hand woven, handcrafted out of materials that will soon degrade in the ocean, materials that stick around for a very long time. And during the time when they are lost or tossed into the ocean, continue extracting wildlife or killing wildlife. So it's the biggest impact that we have had has really occurred, including whaling in the 20th century, and especially the latter part of the 20th century, as markets became global.
Yeah.
There was a time when you could catch a lot of fish from a spawning aggregation, but you didn't because you couldn't consume that many fish locally. But now fish can be packaged into boxes and sent across the ocean refrigerated or by aircraft. They can go from Massachusetts to Japan overnight and be in the fish market the
That's
next.
right. It's that 21st century and 20th century transportation and logistics that are fueling in part along with
But
subsidies.
at the same time, we're lagging behind in exploration. We're still, I mean, fortunately, there are millions of scuba divers that at least get people to put their face and their body down to 100, well, 50
Yeah,
meters.
and I would argue that swimming is key, and we're not doing as much swimming instruction. There are not as many pools, and
Yeah. And
feeling to be unfearful about the ocean is like a key to making.
Most of the ocean
yeah,
is not up here.
Yeah,
And some of the creatures you're describing get their groceries 1,000 meters down.
that's right. We'll get to a little bit of that. So this is kind of a view we have now of a lot of different animals. This is our tuna data set on the right, and then on the west in the Pacific, we've been able with our bigger tagging program to do turtles and whales, tunas, and birds. There's about 26 species represented on that graph. And so, the question now that you know how the tags work is how do you really get them out there in a lot of animals? I'm going to illustrate the tunas first. In the Pacific now, we tag small tunas. In the Atlantic, we tag big tunas. And we actually do a surgery, just in case it wasn't clear. We make a slit, we put a computer inside the animal. The light stock that's going to measure photons is outside there. And then we sew it in. And then we let the animal go. And when we let the animal go, we rely on fishermen to recapture it. The longest out tag now is 7 years of data on
wow.
7 years in a row. And then for Sylvia, this is the 2000th bluefin in the Atlantic tagged a few years back. That's me in my old jacket. And that was something to tag the 2000th fish in the ocean there. When they go out, the way that fishermen know that they're out there is they have a green tag for money. They return the tag. 50% of the bluefin in the Pacific, 900 tags, 50%, no, 1,000 now, have been returned. That's a lot. That's a high mortality. In
Yeah.
the Atlantic, where there's bigger fish being tagged, all these countries have returned tags. This is a tag that was getting returned from Ireland. It's about 25% that have been returned, and that's still pretty high mortality.
Yeah. Yeah.
Yeah.
So there it is. That's the map now, almost up to date of return tags in the Atlantic. It's kind of what you dreamed about. There they are. That's where bluefin are. It's not like we discovered that. The bluefin showed us where they are. And we're now working. We just got the first return. We just heard at lunchtime here in the Black Sea, the first fish
Really?
ever that's gone back to the Black Sea. And it's an amazing story on the tag. So bluefin have been extirpated from this area, so they don't anymore go down here. But as a whole, all those points are made by single tracks through the photon astronomy. So they're all mathematical tracks.
Mhm.
None of this is live tracking. And then that's an example of a tuna going to see where Sylvia was born 2 times, once to the Gulf of Mexico and then comes down and does a second trip. That's pretty hard to get 2 years into the Gulf of Mexico.
Yeah.
Much more common is to get tunas who we tag in the West and they go back to the Med. That's 3 years in a row spawning in the Med. There's another one 4 years in the East after tagging in the West. And that was the beginning where we began to understand European fish who are born in Mediterranean waters and are taken in large mass by Spain, Italy, and France. There actually are tuna. And I want to emphasize this. I haven't shown this in public too many times. We have a new paper coming out in PNAS about this. This is the Gulf in orange. That's the extent of the stock. And now you're going to see the European in white come on top of that. So these are European fish that come and use the shores of North America. Note over here, these are Pacific bluefin tuna, and they're making a 5,000 mile migration. This is about a 2,200 nautical mile migration. And in purple, they go back. And so what the tagging has shown, really before the genomics, is that the tunas are doing what they're doing in the Pacific. They're born in Japan and Taiwan. They come to California for almost 8 years. They go back along the transition zone, and then they spawn on the other side. So here they're born in the Med. They come over, they mix with the Gulf of Mexico, and they go back. And that's a hard problem when 60 nations are trying to catch you for quotas to manage.
Yeah. It's just wild.
So what we've been able to introduce is also with the help of UBC, a new management model. I'm very proud of the effort that went into this. It took years, and it's now actively running at ICAT, the International Commission for the Conservation of Atlantic Tuna. And unlike when Sylvia was younger, it's now led by some of our students. It's also mathematically very advanced. And while it has problems at the management level, do the managers listen to the scientists? It's way more, it's probably in the best of management for bluefin that you're going to imagine, in that they're using a pretty hard model to fit the data to. So they take tagging data, otolith data from the ear bone. It's like a birth certificate. It says if a fish was born in the Gulf of Mexico or the Med. And now, finally coming online with work we're doing in our lab and in other labs around the Atlantic, we have genomic data. So what that means is that if a tuna is tagged in Canada and is thought to be owned by Canada, if you will, it actually isn't a Canadian fish. It's actually moving all the way back to the Med. And in many cases, we can now genomically say it's a Med stock fish. So the models have to start accommodating for this, and they're just doing it now, in which NOAA had managed the tunas in the world as if there were two separate stocks, not mixing. And now what we know is the European stock is actually highly in our waters, and most of the recovery of bluefin is probably the European stock. I'm going to sort of end the bluefin section here by saying what I envision could happen soon, with a little help from the foundations, is that the way in RFMO, that's the UN body that manages a tuna, works. So we have a UN organization in the Atlantic called ICAT, and in the Pacific, there's a couple of them that manage East and West, and one in the Indian Ocean and one in the Southern Ocean. And the Southern Ocean one is quite good. They have a very strong scientific body, what some might say the best. ICAT has gotten very strong, and then the Pacific is weak in its management. And I just want to say one more thing, and I'll turn it back to you. And our vision is that with the genetics, this is our graduate student who just graduated, Chloe Michaels. She's one example of a person who's come to the help of tunas by being able to genomically, with a whole genome, show that the Gulf is different than the Med and blue, that they are distinct divergences. So that means we want to save their lineages, because their lineages are going to offer good proteins and genomic mixing that will enable them to ensure their futures, that we have to manage for their futures, knowing what their genetics are. And that we think that right away, like if tomorrow somebody, wanted to make a business, we'd be the first to join. But more importantly, can we get the world to start genetically testing at the dock? That's what we'd like to see. That day has come.
Yeah.
We talk about managing the fish. We can't manage the fish. We're managing ourselves.
Well, I agree with that. I mean, we're managing humans. And what I'd like to say, Sylvia, is we've come a long way in 25 years.
Yeah.
And we have the power tomorrow. The people who can own the genomic dockside testing will be able to tell everyone what fish you have. And that will be the beginning of clamping down on catching too many of one quota or the next. It's a big day in tunas. And this is not something that's been presented publicly very many times yet. I'm really proud of this. So you could control the tuna from a scientific perspective, and we could, if we had the right foundations and people working together really make what some might call a blockchain of control that then would make it hard for the people who don't have these technologies to understand how to do it. And my dream is a tuna chip in which we've learned across the planet now. This is human fishers in green, longline fishing that we've mapped with our friends from Global Fishing Watch. We
Mhm.
know that soon you'll be able to go to Tokyo or go to any fish market, pick up a tuna, and know from just a genomic screen and a screen of some of what's in it, which ocean, where it came from, what population, and that's pretty powerful.
That's exceptional in the 21st century to be able to really track and know who's who. The question is, to what extent can we really continue to extract wildlife as a source of upscale dining?
Yeah, I think that the answer is straightforward. We need to be producing the animals. If you want to eat tuna, you should be making them in a tank. I'm not against that. I don't think you should be taking them all from the wild. Can we manage them successfully? Maybe. If
Maybe.
we take this mathematics that has come of age, take the tools that have come of age, we just brought them back right across the planet, but it's taken strong leadership and strong science, and there's very little investment. So compared to a salmon where there's probably 2 billion in,
Oh yeah, it's crazy.
yeah.
Do you think that the BBNJ treaty can help to mitigate some of the taking of tuna on the high seas where they just go out with these big refrigerated vessels and just strip mine them, literally out of the and just put them on ice and send them everywhere. Can there be some help with the high seas protections?
I'm not sure yet. It's just brand new. It's obviously terrific. We thank everyone who worked on all of that, and I think we're going to need stronger tools than the BB&J. Like we're going to need tracking devices that help show who the poachers are and things like that. But at
Yeah.
the same time, I'm hoping that this coming of age of the tools will change the game. To some extent, people investing in these technologies to make it to the end game, like COVID tests for tuna, they will really own a lot if they can
Yeah.
get there. So
What about,
I've shown you this map, and I want to say I've only shown you the tuna side. I want to just take 1 second and show you the billfish in white. So they're the understudied animal. So tunas have thousands of days on these maps, and billfish are just coming of age. They're beautiful animals. They're iconic, like the black marlin. I just started studying that with the Guy Harvey Foundation, and they don't get their due. So yellow again are tunas, red are the sharks. So you can see where our tags have been.
Mhm.
And there's a gorgeous picture for both of you, taken by Guy's daughter, Jess Harvey. That's a black marlin that we just tagged. And these are the pop-up tags. And one of the challenges for instrumentation on a marlin is we can't put the extra loop on that stabilizes it on a tuna because we do it on the deck. And so
Mhm.
these things are sort of moving around, the antenna hits the fish, and it's been a challenge keeping them on these big, iconic fish. And so we're
Yeah.
just getting the first really big tracks. This is a really nice display of billfish that IGFA and our lab have put together. Our lab's doing all the track analysis, and it's just where blue marlin and black marlin go. We're getting there. Took a while to get this kind of data. And this is a big citizen science program. So sport fishers can purchase a tag from IGFA or sponsor a tag. We like to say, note the gaps we have in the Indian Ocean right
Oh
now.
yeah.
And yet, around the planet, fishers are doing this. And so they're helping scientists by sponsoring tags and then putting them out there. And I'm sure the data set will get better. But that's what we've started as a new project with our friends at IGFA. And so I just love seeing, here's the Pacific. I think the longest black marlin in our tracks might be this one right here. And you realize how big the Pacific is. That's a 5,500-mile track. Isn't that amazing?
That is incredible.
And then for marlin, this is blue marlin, my favorite. I've done a lot of my career tagging blue marlin in Hawaii, and
Hmm.
they tend to go towards Tahiti and towards the Eastern Pacific. But I want everyone to notice where the EEZs are or where these puffy circles are. And these puffy circles are protecting, of course, the EEZ of nations. But notice how the marlin are in the middle of where there is no protection. And so
Yeah,
that's
the
really,
high seas, right?
they're in the high seas, and that's
Yeah.
a big problem.
Yeah. Yeah. It's a huge problem, and I'm really hopeful we can
Right
get
here,
some.
down to the scattering zone.
Yeah.
Yeah, so here's this is also the new data coming from Marlin, worked up by a beautiful effort by Sammy Andriachik in my lab, where you can see the marlin goes deep during the day into the Sylvia's mesopelagic and then comes up on the surface at night that goes deep during the day. That's a pretty, no matter where you tag a blue marlin, you're going to get the same track. We've done that around the planet. And then if you look at the detail that you get from our tags, you can see the cold temperatures in light blue. You can see the surface behavior. So you might be looking for a mahi up here, but the animal's definitely going on a V dive into the where the it's not lit very well. Not sure what they're seeking down there. We haven't really figured that out, but as you said, Sylvia, they're using that area.
Yeah.
We need to
So
get.
our work in the Pacific has been extensive. I'm not going to detail it. Pacific bluefin tuna, some seal work by Dan Costa's team. We're the shark guys who have basically put out all the tags on blue are the salmon sharks, black is the white shark, going to the White Shark Cafe, red is the makos, and then they're taking lots of data. Here's George's leatherback work.
Oh,
And
yeah.
what I want to get to is, if I can, some white shark data. So we were surprised in our West Coast project that if you tag any of the animals on the West Coast, they show us right away, this is the salmon shark up here, that the habitat is the coast for part of the year and then into the subtropics for part of the year. And that was a big surprise when we found this out. So these are all the sharks of the West Coast, the salmon shark, the laminate sharks, the white sharks, and the makos all together. And in a seasonal cycle, we're able to see that they'll come down in the winter time, right about now, February 25th. They're all in the subtropical Pacific here. So that means we have to save sharks, protect these regions, and
Mhm.
pay attention to them. And then the makos come down to where the yellowfin get caught, and they often are bycatch. Here's the white sharks out in the middle of the White Shark Cafe. So what I just showed you was the salmon sharks coming all the way down. It's an amazing story. This is a very unknown shark. It's up in Alaska. It's the most endothermic. It makes the most heat. That's why I like it. And then it can carry a satellite tag. So now we're going to talk directly to satellites.
Right.
Then it's doing what Sylvia was talking about. It's at the surface right here in the warm temperatures, and then it goes down half a day, even all day long. It'll be down deep looking for probably something down there, squid, pollock, who knows, and then comes
Yeah.
back up. So that's incredible biology. And of course, there's the white shark. This is unpublished data so far, even though it should be published by now. It's from our big effort here. We're trying to get this to a big journal shortly. And what we know is if we tag a white shark on the American West Coast here, they all go to the same place. There's a gathering place. We call it the White Shark Cafe. And you don't have to put too many more tags out. They're all going to go there. So
I've briefly noticed. Yeah.
yeah, and then when you look from the surface, there's nothing there. And the story is in a paper that's about to be submitted. So I'm not going to tell it all today, but there they are off in the White Shark Cafe. We went there with the Schmidt ship. And then they're also off in Hawaii, the
Mhm.
white sharks of the American West Coast. Now, here's Dan Costa's elephant seals, and they're quite a bit north of the white sharks. So the white sharks aren't chasing elephant seals. So I just showed you the white shark biology over here.
Yep.
And some people thought they were going offshore to get these guys, who unfortunately, many of you just heard have got a little bit of the avian flu here.
Yeah. That's really going to be devastating.
But this is amazing that they're way up here and the white sharks are down further to the south. So when they come home to California, we've put cameras on the animals, and here's an elephant seal who hugs the bottom. They're kind of, you feel the fear they have for the white sharks near
Oh,
Anya.
yeah.
Here's some sea lions with a bite mark. And as some of you know, we've set up camp at Año in the Farallones for over 25 years, where we're doing something unique. We're putting in acoustic receivers, and we put acoustic tags on the white sharks. And the white sharks swim by the buoys, and they send data to the buoys, and they tell us in live real time that they're there. And because of the Ocean Protection Council, some of our taxpayer dollars are helping us in California, and that this is the Monterey Bay National Marine Sanctuary. This is a place larger than both Yellowstone and Yosemite combined. In our new project called the Monterey Bay White Sharks MontereyBayWhiteSharks.org. You can go in and you can pick up the live receivers in orange. There are a
Mhm.
lot in Monterey. And just a quick look down on our ship, that's our boat with a drone. There's our white sharks around our little 24-foot Grady. And we take a picture of them with a GoPro to get their ID. We measure them with a drone. We look at the sex with the female there and the male here with the GoPro. And then every shark has a unique dorsal fin that we keep track of with a computational program. And then we put a tag on it. That's an acoustic tag. And then Shiley, Jory, and Raksha in our lab are doing the genomes of the white sharks with Beth Shapiro's lab. And then we're also doing eDNA. And so we'll tag a fish under the boat. And that acoustic tag now will talk right to the buoy, 500 meters distance is the max. And then you get data where every number is a white shark. The white shark is talking to us saying, I'm here, I'm here, I'm here. Then the gap is they go to the cafe and then they come back. And so this is today. I just took this off the web. We
Cool.
had the white sharks all left suddenly on the 27th or so of February. And the only thing I've noticed is I think the whale watch boat said they saw some Biggs killer whales.
Yeah,
So
I was gonna ask about this
yeah,
interaction that's been seeing. I guess maybe it's always happened, but it's just been really
yeah.
noticed.
I
When
think that's all of
The
our cameras are getting those pictures, but it is a strange story, this leaving. These are the sharks we just tagged this year. And we had quite a group here. I was out for the day that we tagged these. And then of course they're gone now and just a few are left. So that's cool stuff though. But what it does is it gives us with the acoustics the coming and going. So this would be the start when we tagged and then they come back and we add a few more. The coming and going, the coming and going. And we can do what we call mark recapture models and count some of the white sharks that way. We count them from the pictures. We can also run a wave glider up the coast.
Mhm.
We did that. And then find a few stragglers. These are orange or white sharks. And if you see a little peak of land and there's a few marine mammals around, I'll tell you, you find one of those white sharks we haven't seen in a while, and it's kind of interesting. And so then if you come back and you go around the Farallones, you see quickly, don't go swimming anytime
Oh
near
yeah,
the
I love that
Farallones.
area.
And there's like all these different white sharks. Look at the numbers here. It's just crazy. Those are all different individuals. Like, anyone who's planning to swim the Farallones, like, don't do it in October, November, December.
Right? Yeah, the Sharktober months.
Yeah, it's a Sharktober. So that's what it looks like. We use a decoy. We use a slight bit of bait. It's actually over here. It's very regulated. It's not in the water. All this is only done at Año Nuevo.
Mhm.
And we did that part. And here's a couple of really great videos of what it looks like. So that's Taylor Chappell and myself working a white shark. This is a big shark, one of the biggest ones we've seen. It was coming towards the boat, kind of grabbed the decoy. It wasn't supposed to do that. I was the fisher who should have snatched it away. And then once we get our rhythm down, we can bring this shark right to the boat. Same shark. We tag it. And that's how we're doing this. And what we're proud of, it's all uninvasive. We don't really pick the shark up. We don't put it on a platform. We don't do any of that. It's
Yeah.
all done with a swim by.
Yeah, and that's great because isn't it true that because they're such big animals, they have such
and
a
the liver.
the big liver that if you were to kind of haul them out, that you have the potential to kind of get
Yeah.
damage from
I think
gravity.
our colleagues at OSearch have shown that they can be done. It's not something I wanted to deal with early on, so our program established the uninvasive way of doing it. But
Yeah.
even though there was cautionary concern about picking them up, they've done a fantastic job at their website. You can see their sharks at our website. They're on the East Coast, we're on the West Coast, and everybody is doing wonderful work to help us understand the white shark dynamics. For the West Coast, in case you're interested in going swimming down by Anya, this is October. That's why it's called Sharktober. And the blue are the males, and the orange are the females. And you can just see there's a peak right here. And then the females kind of come a little later. So let's do that again. Adult blue. There's the peak. And that peak is right at October. And then the second peak is more towards December, as if the females might be looking at the bigger elephant seals. We think the males, some of them are taking the sea lions, and some of the others are taking the elephant seals.
Yeah.
Okay,
Yeah.
I'm going to wind up with just a few interesting points I want to I want to bring home. It's
All right.
a bit of a debate, so let's do it real quick. So here's the protection at the sanctuary, and here's the EZ, and these are white sharks. And the white sharks go all the way out to the monument. That's protection. So if you look at the green, there's very little protected area for white sharks because they live in the subtropical gyre. And here's the White Shark Cafe. Oops, let's get rid of that. That was a little bit of a mix up there. So when they come here, their protection in white for white sharks, in orange for salmon sharks, is the Monterey Bay National Marine Sanctuary. All right, so the area that's protected is quite small, even though it looks pretty big. These are bluefin offshore. They're more on offshore fish. And even though we have all this technology, we're not really protecting them. So the way that we're working to protect them is we count their fins from the unique ID that they give us. And then a nice team at UCSC led by Dr. Couture and her graduate students have helped us use computer vision and AI. And now we can come in from the field, throw in a fin, match it to anything that's in our database, and then immediately know which are matches and which are newcomers. About every year, it's 50% or so are newcomers, 50% are sharks who've been here before. And that's how we keep track of them. Graduate student working with the aquarium and our team, Paul Kanive, who now runs the California White Shark Program. He's made an estimate. We're working on a new one. They're working on updating their model. And it says there's about 260, that's 250 right there, adult white sharks in the Central Coast region. So no matter what, it's not very many. There's
Under
probably
300.
Under 300, yeah.
Yeah. So you can't afford to lose too many.
No.
So I'm going to just sort of bring us into the sad stuff right now. So white sharks are captured. It's a big debate. It's not too hard to find them in the Taiwanese longline industry. We have friends over there who pay attention. We know that in this region where the white sharks are, there's intensive longlining.
Yeah.
That longlining, this is just effort by reported by one of the RFMOs. We're up here, does exist. I'm not accusing anyone of doing it, but I'm saying it does exist. We've been told that when they catch tunas, the white sharks almost see it like a lollipop and get stuck on it. They do have to release them, but we do know sharks show up in the markets.
Sure they do. It's really
So we've
sad.
done a lot of papers. It's been a bit of a debate, so I'm not going to go either way in the region. It's hard to kind of pin what's going on, but we can look at who are the users of the region. Our graduate student, now an employee, of course, of Global Fishing Watch, Tim White, did a beautiful study where he looked at these regions, the region around Hawaii, the region around the cafe, and along with a second study, they mapped what's the overlap. And there's no question that we're concerned. So I'm going to end on a little bit of hope. So I've been thinking for about 7 years, how do we go forward, knowing that the high seas are a challenge? And the idea that I came up with, that engineers here at Stanford helped to bring to fruition, is could we put a tag on a shark, on its fin, and we call it the Fin Alert Shark Tag, that could connect in seconds to Iridium after getting a GPS fix. So it takes time to get one GPS fix, and then it has to talk and give us the information through Iridium. That's very different than doing things with geolocation. So the idea is, if this is Global Fishing Watch data, now looking down on one of our favorite spots, the Galapagos, this is the island area, and all of these colors are fishermen. These are humans. For only a period of April 30, 2024 through July 30, 2024. So it's not a very big period, and that's the effort that's going on around the Galapagos.
That's insane.
But at least it does suggest that they're respecting, to some extent, an area that is legally protected. Look at the space around the Galapagos.
Yeah.
Yeah.
But it's not a very big space. So like if you're a shark, are you going to stay in that space, or are you going to come out?
No, that's like the whale shark Hope that did have a tag on it.
Right.
And people were watching it, and it was hanging out at Galapagos, and then it took off towards Cocos, and then just like
Boom.
gone.
Yeah.
Right. That happens a lot with our tagging. So I was inspired when I was working with the Bertarelli Foundation over in the Chagos Archipelago, that's now been given back to Mauritius, that no matter what we were doing, sharks were being killed right in front of us. So it was a protected area as big as it gets, France, the size of France. And we have an army base a military base there, and yet, fishers come in and take the sharks.
Yeah.
So the tag is intended to be off-the-shelf components and cheap. And so we
Mhm.
built some early models. Think of it as like we go to the store, such as Macy's, most of us
Yeah.
won't steal, and that's because we have a fear of being caught. So we have tagged the clothes. And so we made the first and second generation tag. We had to get through a lot of problems. It turned out to be much harder to use GPS than the engineers thought. Imagine that.
Yeah.
And for everyone who's listening, once you get below the water, the ocean, you can't send a radio signal. And that's what these tags are doing. They're sending a radio signal to GPS. You use your phone. Everyone's got an intuitive sense of how GPS works. The
Yeah.
phone sends out, a message, and the satellites hear it.
Yeah.
So we made the tags. They were a little bit big. They weren't small. But the best part is we put them on our test friends. And so we used Dan Costa's elephant seals, and they found out right away. They were some of the best tracked seals. They went out and back. I don't know if I have the data here, but they went out and back. Oh, here we go. And what Dan loved the most, and the team of his students loved the most, was how accurate the tracks were. Like no one had seen this accuracy. This was just recently, and now what we're doing is we're going to put them on sharks. So we've already done it. We put the first one on a porbeagle. It worked until it went down to North Carolina. And it seems like somebody ate them, which is
Oh,
kind of the bad luck that happens when you're a researcher putting out a fast tag.
yeah.
And
It's
then we're
like the Garmin InReach, that we use for scuba and hiking and all
yeah.
the other applications that has is like that's like the InReach for sharks. You get in trouble and it sends an alert.
That's exactly what this is. We did try to get Garmin to work with us, where we're, in our opinion, doing better than the inReach is we're down to
Yeah.
1,000 meters now. So we
Yeah.
can use
Excellent.
these devices. We hope the last 3 years, and we did get a nice grant from the Schmidt Foundation to help us test in Galapagos. We're going to be going to Malpelo. We're going to put them on white sharks this year. And then we're trying to design some that are more streamlined for the tunas and the billfishes. So that's pretty much the story. I just wanted to end today, from my perspective, just thanking everyone, all the students, the postdocs, and the foundations that have helped made all this work possible, and open it up for questions.
That's excellent. Thank
So
you
I
so
have
much.
a question before we go elsewhere. So all your tagging information, how can that be used to gauge population decline or increase?
Ah, good question. A nice, good question. In the Proceedings of National Academy paper that we just are publishing, we bring to life a study that is done with acoustic tags that we published in the in other journals, and now this is a review paper that we've done. And what we did was with the tunas is we put their tags on, and they cross a gate that Canada built underneath the sea, and it's built by the Ocean Tracking Network, a very big program that's an outgrowth of the Census of Marine Life. So after we finished the Census of Marine Life, Canada took the lead on a beautiful project that laid this underwater receiver network, and the tunas cross it. And if the tag stays on for 5 to 10 years, what we can do is get a moment when the tuna leaves the North American coast. So some of the leaving of the North American coast is natural mortality, and some of it is fisheries mortality. And we're working on how to do that best with both genomics also included now for stocks. And what you get out of that is the first estimates of natural mortality, which are critical for building population models. The second way is with censusing with numbers. That's what we do with the white sharks. When they go and they come back, they go and they come back. We combine that. We're combining it now with the fin
Mhm.
census. So you have two mark recapture components. So that helps you do the census. And then in all cases, this is used in management.
Yeah.
Well, from the 1950s, when industrial fishing, commercial fishing, even in coastal waters, really began to scale up with new markets and new technologies to find, capture, and send fish from anywhere to anywhere else. What the numbers that NOAA in 1990 had suggested that for bluefins, literally 90% had been taken from 1970 to 1990 of the bluefins in the North Atlantic. And numbers in the Pacific seem to be even more drastic in terms of the speed of decline from where they had been in a very short time, brought down to remarkably low levels. But there are still bluefin. So we could allegedly take them all if we were really inclined to do so. So some of the policies for trying to at least have some of these big fish out there seem to at least not take all of the fish, but their
Yeah,
numbers are
I mean, a quick way to summarize it is there was a bad period. We've been through it, and you and others influenced a whole generation of young leaders. They've risen up. They've worked with us, the scientists, and as a team across the planet, we've brought the bluefin tuna back. There isn't a population that's currently considered threatened or endangered or anything. The bluefin are doing well. The only population that might have a little bit of problem is the Gulf of Mexico. I think it's not clear yet, but it's where NOAA's put in the most work to use something called close kin mark recapture. So off the, almost like the sky's the limit, you can now, because of the small population that humans did reduce, the bluefin to, you can now count their parents. So you basically pick up a bluefin and instead of tagging it,
right,
you can figure out from genomics and the markers who
from
mom and dad were. And
Wow.
there are some pretty darn good mathematicians out there working together. And what they're able to do is figure out how big the population had to be to make this many genetic pairs.
with the
And
tag.
that's amazing.
That's a
Yeah,
huge plus.
yeah.
Yeah.
But there's some recovery, but nothing like back to the 1950s
No,
level.
I would agree with that. I would say, you'd have to say, just to be careful there, the 1920s. And the recovery in the East is big, Sylvia. It's not a small recovery. It's big. Like
Good.
even we can't figure out how it got so big. But the bottom line is there's a lot of Mediterranean bluefin everywhere. I don't think the Gulf of Mexico, we have an answer yet that is solid, and I'm not going to comment on that too hard. The Pacific bluefin was definitely demolished, but guess what? It came back too, and that's what led to the last decade of high bluefin here. And so here's what's happened here. It's kind of a good and a bad story at the same time. A lot of us experienced the story of large Pacific bluefin. We didn't even know they were here really when it started. And then, of course, we've taken advantage of that to put them in captivity. It got pretty amazing, like fish as big as, spawning age fish, and then they go back. So the glory years were up to 2016, I think, 2017, and it even was okay in 2018. But the quotas did go up, and those quotas going up is impacting the California fisheries. So the California fishery is a hard one to understand. It's simple if you think about it, but it's a great story, so I have to tell it. It's the largest migration of a fish on the planet. The tuna is born in Taiwan, swims in year 2 to California, spends 6 years going north and south on our side, getting bigger, goes back and spawns probably by 8 or 9. And then there's a post-spawning migration to New Zealand.
Wow.
And that's a big one that no one's been able to capture. We've tried. And then after it's in New Zealand, it comes back. We have gone to New Zealand 3 times, tagged bluefin, and the coolest recent story is we just had a fisher return a 7-year archival tag. We
Nice.
thought it was the gold mine, this is it. And the long awaited I can retire kind of tag. And what happened was they got us the tag and they sent us a picture. And I remember looking at the picture saying, wait, the tuna's been out 7 years and it's only that big. And I said, that's not a Pacific bluefin then. So it turned out it was a southern bluefin, and none of us had ever tagged a southern bluefin. And no one, as far as I know, has had a spawning migration on a southern bluefin. So what we have in our hands is a fish who went from New Zealand around Australia to Indonesia 7 times.
Wow.
And every year it went. So there's been a huge, oh, do they skip spawning? Do they don't? And most people can only put a tag on for a year. So they all say they skip spawning, when they don't spawn. When I say, no, they weren't big enough.
Yeah.
They spawn too soon. And the 7-year archival tag is the longest record I've seen. It is an amazing record. And we didn't think it was all there because the stock broke pretty quickly. And when the outside stock breaks, you lose light. So you can't see sunrise and sunset, but you can
Right.
get longitude. And we
Yeah.
have longitude on the tag, but we don't have latitude. That's a long story we won't tell today. But the bottom line is 7 years in a row it goes to spawn on the exact same day. Like,
That's incredible.
and
That's
that means that these animals have clocks better than ours. It's just like us, sort of.
The next big question is, okay, so you have these big fish, they have to eat something to stay
big,
alive.
bigger.
And the market now and the capture now of high seas squid
Oh yeah.
and going into this migrating zone of life that, that is
Yeah. No, I get what you're saying. One of the projects I'm hoping to do, depending on my student, is I think the animals can tell us where the carbon is in the ocean, in that we've been going up and down with animals for a long time across the planet. You can see that in the data I just showed you. And in the tag, when a light sensor that has a beautiful 9-decade light sensor, these are good light sensors, when they go down and the water is scattering particles, when they go through the DSL, we could see it. We tested this out on trips with Stanford students. So could we actually measure where the DSL is across the planet? And the White Shark showed us the White Shark Cafe. Hopefully soon you can read a paper about it. The bluefin, we've already published the paper, tagged in Ireland and Norway. I should have had that slide in here, but I didn't. Some of the greatest tracks on the planet. They kept going to a place none of us knew existed. And where was that place? Can you guess? We're talking about the biggest tunas on the planet.
Don't
They
know.
went to the Newfoundland Basin, like in the middle of the ocean. Like, who knows what's
Yeah.
in the Newfoundland Basin? Well,
their cafe.
Yeah.
yeah, so it turned out it was the
There.
Atlantic Cafe and the Pacific, and the postdoc looked up what's there, and there had been a couple of cruises, and down at depth, you've been to a market, I'm sure in Madeira and other places where you see all these silvery trichiurids, snake mackerels, all sorts of things. So, there is a whole carbon sink down there that the tunas know about. And I just think I like to kid my marine carbon community and say, is anyone counting how much carbon's in a tuna? And like, if we left the tunas in the ocean, we could think of them as vessels of
They,
carbon.
they are biodiversity and tuna and carbon credits.
Yeah,
Absolutely.
If whales have a carbon value of $1 trillion, as has
Alive.
been
Yeah.
live tunas, I mean, live,
Live whales.
what's the carbon value of tunas alive?
I think it's high, and I think no one's calculated it. We need to start doing that.
Yeah.
I also think that I mean, I'm not the expert in the area, but I really think we don't have the mesopelagic mapped right. So I think that there's a lot of carbon there. I've been studying it with tunas and with students forever, and it's kind of misunderstood. And I think that carbon that's moving up and down, while we think we know where it is, I was surprised on our recent trip on Stanford at Sea. We went to French Polynesia, all the way through the Tuamotus, all the way down to Mangareva. And for the first time, I saw the DSL disappear. So
Really?
yeah, when we're taught that it's an oligotrophic Pacific, there is this big dome of hot water there, salty hot water. Not many people get to the gyre of the Pacific. And we were expecting a desert, so we did dress up for a desert day, and so it was a desert. And I think we had some nice on deck discussions about why does NASA put the space junk there? And after we sailed for 1,100 nautical miles through a place with nothing, we did come out realizing, wow, it's pretty empty here.
Wild.
So I think that's been a big lesson, that there are barriers out there. There are places that are not as uniquely rich as the North Atlantic. And I think we have to do a better job of mapping this planet sooner so we understand what we're doing, and so we get the calculation and the estimations right on the map.
Get the math right.
Yeah.
Exactly. Well, we've crept past the top or bottom of the hour.
Oh my goodness.
Um, and I guess we had one question, but we can answer that offline in respect for your time, because I know you've got a hard stop, Barbara. But I do want to say thank you so much, and thank you all to all of our viewers, and of course to the Ocean Elders. And Barbara, thank you again for spending all this time with us today. It's been really informative, and I hope it's stimulating a lot of questions out there for people and answers. And yeah, so thank you for showing up and participating with us on Dive In.
We
Water connects us all.
we owe each other an expedition together.
Yeah, I know that. And thank you. I owed Sylvia a talk because she's come at a drop of a hat and inspired probably 4 of my classes, and very memorable. I want to tell everyone there is a brand new film we just did. It's a short film done by one of Sylvia's favorite people, Kip Evans.
Huh?
It's on our website, montereybaywhitesharks.org. We put it up 3 months ago. It's got 250,000 views. It's cute. It's nice. It tells the story of West Coast white sharks that we're also helping to protect. And I hope you all go take a view of it.
Yeah, and Gigi just put it in the chat. So take good care. We will see you next month. And remember, take care of the ocean.
As if your life depends on it, because
It
it does.
does. Thanks, everyone. We'll see you again soon.